Good morning, everyone. I'm Thomas Provence, a PhD student of the University of Limoges, and I will present you now a part of my final results of my thesis. During this presentation, I will talk about the development of a microsystem that can sort biological cells based on a phenomenon named dielectrophoresis. Along this presentation, I will talk about the objectives of this microsystem. I will next develop about the phenomenon used in our sorting system. With the dielectrophoresis phenomenon, different parts of the cell dielectric characteristic can be focused, and so I will show you how we can probe their internal dielectric properties. Based on this knowledge, I can show you how the sorting system works and some experiments showing the impact of the sorting frequency used. And finally, the importance of the condition to modulate the refinement of a population of cells. And I will conclude this presentation. In our work, we aim to develop a tool for the biological field, which can be used for therapeutic or research use. This tool will sort cells based on their intracellular directorial properties, which are linked to the biological characteristics of the cells. The dielectrophoresis phenomenon is a contactless force that will interact with biological cells and will be in interaction with fluidic force in a lab chip. A lab chip is a combination of microsystems like horse, which can. A lab chip is a combination of microsystems like ore and a few millimeters. The channels in which the cells will be flowing are about few hundreds micrometer, and so the microsystem is at the scale of the cells, which are about, in our case, around 16 micrometer. And so the scaling permits us to exploit different type of force, like acoustic, optics, and in our case, the dielectrophoresis force. Between two electrodes, if we apply a radio frequency signal, a non-uniform electric field will be generated. In this electric field, a cell will be polarized, and so a force will be applied on a cell. The force will be linked to the gradient of this electric field, as shown in this figure. It will also depend on the properties of the cell, like its size or its Clausius-Mossetti factor, which are linked to the dielectric properties of the cells. Depending on this Clausius-Mossetti factor, and so, as shown on the photography, cells will be attracted around the edge of the electrodes. In other case, if this Clausius-Mossetti factor is not positive but negative, cells will be repulsed by the electric field. And this dielectrophoresis force is more intense around the edge of the electrode. As I said, the Clausius-Mossetti factor relies on the dielectric characteristics of the cell, and it also depends on the frequency of the signal that generates the electric field. This is the evolution of the Clausius-Mossetti factor depending on the frequency of the signal that generates the electric field. The blue curve is the real part of the Clausius-Mossetti factor, and the orange one, the imaginary part of the Clausius-Mossetti. In our case, the intensity and the direction of the dielectrophoresis force is only linked to the real part of the Clausius-Mossetti factor, and so the blue curve. If some cells present different membrane properties, it will be at this range of frequency that the Clausius-Mossetti factor value will vary the most, mostly at low frequency range. Now, if some cells present different internal dielectric properties, it will be at this range of frequency that the Clausius-Mossetti factor will vary. The impact of the internal dielectric properties mostly start when the cell begin to be attracted by the electric field and continue with higher frequencies. So we observe that at this particular range of frequency, both membrane and internal dielectric properties of the cells impact the Clausius-Mossotti factor. In our case, we aim to sort cells only based on their intracellular dielectric properties, and so we will focus on the UHF frequency range. To be particular, it will be with frequencies higher than the crossover frequency of the cells. The crossover frequencies are particular frequency where the Clausius-Mossotti factor and so the Dielectrophorosis force are null. And so, in our experiments, we only use repulsive behavior. This is our sorting system, with 2 inlets at the right and 4 outlets at the left. At the center is the sorting area, which presents 2 golden electrodes that will generate the electric field. The cells will come from in a suspension in a low-conductive medium to the sorting area and will be focused by the second inlet, which injects the same medium. As I said, between 2 electrodes, an electric field is generated, and the dielectrophoresis force depends on the gradient of the electric field. The area where the cell will be the most repulsed by the electric field are these 2 red areas around the edge of the electrode. This red area will act as a barrier that will deflect the cells if the cells have a sufficient strong repulsion behavior from the electric field. And if the cell isn't sufficiently repulsed by the electric field, it will pass through the electric field and be collected by one of the 4 outlets. As you can see, the electrodes present 3 different angles of throw. And for each, the balance between the dielectrophoresis force and the Friedrich force will be different. The first angle is about 35 degrees, and it will be easier to cells to be deflected by this first angle than the second one of 45 degrees. And it will be also easier for cells to be deflected by the second angle than the third one, which presents an angle of 60 degrees. All the cells which have a strong repulsion intensity will be collected by the fourth outlet, and the weakest repulsion intensity will be collected by the first outlet. And as you can see on this photography, depending on the intensity of repulsion that cells present, that will be collected in different outlets. And so a population which presents an heterogeneity in their dielectric properties will be sorted in 4 different subpopulations which present more similarities. I will show you now a short video about one of our experiments. As you can see on the video, the cells are flowing from the right to the left, and when they are around the edge of the electrode, where the electric field is the most intense, they can be deflected by the dielectrophoresis force. And some of them weakly react to the electric field and so pass through the electric field and are collected by one of the 4 outlets. I present you now the population of cells that will be used in the different experiments that I will show you. This is a population of glioblastoma cells which are named U87. This population have been selected for its substantial biological heterogeneity. And so, when we measure the crossover frequencies of this population of cells, we observe the same heterogeneity in their crossover frequencies, and so in their dielectric properties. And so, based on this large heterogeneity of their dielectric properties, this population is good to show the capability of our system. This diagram presents the crossover frequencies of the whole population, and between each color, purple, blue, green, red, and brown, we will have a fourth of the whole population. And so, if we use a sorting frequency at the green line, and half of the population will act with repulsion behavior, and the other half of the population will act with attraction behavior. So we will use higher frequency than the purple line. Based on these crossover frequencies, we can expect for the whole population this evolution of the Clausius-Mossotti factor, depending on the frequency of the electric field. As explained earlier, for each angle of our electrodes, a minimal intensity of repulsion is needed. And so for a fixed sorting condition, the intensity of the dielectrophoresis force will vary between cells based on their Clausius-Mossotti factor. And so each cell that presents higher Clausius-Mossotti factor value than the blue line will pass through the electric field and be collected by the first outlet. All cells that present a value lowest than this blue line will be deflected by the first angle, but if the value is still higher than the green line, the cell will be collected by the second outlet. And if the cell can be deflected by the second angle, if the cell presents a value higher than the yellow line, it will be collected by the third outlet. And for each cell that present a Clausius-Mossotti factor value lower than the yellow line will be deflected all along the three angles of the electrodes. For our experiments, we will only use frequencies between 250 MHz and 500 MHz. So all cells will be repulsed by the electric field. And depending on the frequency used during our sorting, the proportion of the cell in each outlet will differ. For example, at the lowest frequency used, more cells will be collected in the first outlet than if we use the highest frequency. On the contrary, for 500 MHz, more cells may be collected in the fourth outlet than at the 250 MHz. Now I will show you some results of our sorting experiments. If for different experiments we keep the same electric and fluidic condition and only vary the sorting frequency, we obtain these results. For each condition, more than 100 cells are being observed, and the statistical error are represented on the graph. If we focus on the cells collected by the first outlet, we can see that if we lower the sorting frequency, we can see that more cells are collected in the first outlet. And so at lower frequencies, as expected, cells present a lowest value of the Clausius-Mossotti factor than at higher frequency. And now, if we focus on the fourth outlet, which collects all the cells that present a strong repulsion from the electric field, we can see that with higher frequency, more cells are collected, and so more cells present a strong Clausius-Mossotti value. The tendency of the second and the third outlet vary a lot, and there are more certain. The variation of cells collected in the second and the third outlets are less predictable and are more linked to the repetition of the dielectric properties of the cell. And so we can use these both outlets to isolate a particular part of the population. These six collections are for given fluidics and electric intensity. And now we will see the impact of the electric field intensity if we change it. So for given condition and a fixed sorting frequency, we can obtain this refinement of the population. And if we change the intensity of the electric field, we will change the critical value that lead a cell to be deflected or not by the electric field for each angle. So if we increase the electric field, it will be more easier for cells to be deflected by the electric field, and so more cells can be collected in the fourth outlet than weakest electric field. And as you can see in this different collection, without an electric field, all cells are collected in the first outlet. And if we apply an electric field, some cells start to be deflected by the electric field. And if we increase the input of the radio frequency power, the cells are more easily deflected by the electric field, and so more cells goes to other outlets than the first one. So this is the evolution of the refinement of the population if we vary the condition of the input radio frequency power with the same signal frequency and the same flow speed. If now for different sorting, we keep the same sorting frequency and with the same input power, and we vary the flow speed. We observe this different refinement of the population. With a high flow speed, the refinement of the population and subpopulation will be similar than a collection without signal power. But if we lower the flow speed with the same signal intensity, more easily the cells will react to the electric field and being deflected to other outlets than the first one. Whereas decreasing the signal input lead to less cells being deflected to the fourth outlet, with the decrease of the flow speed, more cells are deflected to the fourth outlet. During this presentation, I show you a continual sorting microsystem that can sort cells based on their dielectrical properties in a continuous flow. The refinement of a given population can be tuned by different conditions of sorting, like the frequency of the signal, the input of the signal, and the speed of the flow. Before finish this presentation, I would like to thank the European Union for funding the SUMCASTEC project with the Horizon 2020 program. And I thank you for your attention.